one of the most recent that has probably most national and international sort of interactions is that associated with the Deepwater Horizon spill. And so what I'd like to do in the short time I have is just give some of the information that we gleaned from some of our studies, not the gene studies or anything regarding that, but some of the studies that we did that have basic information regarding exposure, transport of the material from the spill site, and other information as it relates to development and fish development. So I'd like to tell that story, a short story, and proceed along that way. As most of you remember and have had interaction with, the Deepwater Horizon spill a number of years ago in 2010 was a very eventful thing in Louisiana from the standpoint of environmental and economic consequences. And of those things, a number of important features are notable. First of all, the loss of life, the magnitude of the spill, the depth of the spill, issues associated with distance from the shore as related to the depth associated with the drilling incident, the environment itself, and the type of oil. And all of these things sort of intermingled to give a fairly unique profile associated with that particular issue. And so I'm just going to give a few review items here to just sort of put into context some of the research that I would like to present to you. First of all, the size of the spill was over 2 million gallons released, 320 miles of shoreline. You can see areas of heavy, moderate, and light spills, large part of the Gulf. The source of the oil and the wellhead is in that black bolded area. Closest component associated with the spill was 40 miles offshore. A lot of the spills, including the one you heard about in California, was onshore or near shore. This is quite a bit offshore. And it was in a warm subtropical. Now, most of our information from a toxicological standpoint is with the Exxon Valdez, which obviously the water was not subtropical and off of Alaska and had other issues associated with it. All of those things really gave a unique character to the spill. I mean, we hear about the depth, we hear about curtailing the leakage at the wellhead and all those things, but it also did certain things to a unique oil component. Oil, whenever there's a spill, undergoes weathering. And in large measure, the weathering component, we see a loss of volatiles and various components to oil. Oil being a very complex mixture of many things. So we have evaporation and loss of things. We have also material being biodegraded by microbes in the aquatic environment, material sedimenting out, and of course the spread of the oil spill. What was interesting here is that another feature was added to this, the fact that the oil was also brought into solution by the addition of surfactants, or the corexit dispersant. And so we find that a fair amount was brought into the water column, which would affect pelagic or free-swimming animals 40 miles offshore, animals that would breed offshore or otherwise have some significant part of their life cycle. Now, that was done on purpose to prevent damage coming ashore to a lot of the vital marshes in Louisiana. But in fact, a number and a large amount of the material wasn't solubilized, but formed emulsified materials. And that's what the topic of what I'm going to be talking about is emulsified oil per se. Again, oil is very complex, extremely complex. And all I have this slide up here is to show of alkanes, the total concentration of alkanes in there. It's 8,200 plus nanograms per milligram of oil. The aromatic, the PAHs, which are thought to be responsible for a lot of the developmental problems with oil, was around 1,600. And there's hundreds to thousands of compounds in the oil per se. But oil did, in the form of emulsified material, made that journey of 40 miles through the water column. This wasn't oil that was solubilized. It wasn't oil that was affected by the corexit or surfactant, but it was oil that entrained water or went around water to form an emulsification. And characteristic emulsification is here shown with some marsh grasses. There's this reddish-brown material. And there are a lot of different types of water emulsions. If you take oil, vegetable oil, and put it in water, what happens? It separates out dramatically. But there are other types, and this is an unstable type of emulsion. It basically dissociates directly. We also find entrained water with heavy viscous oils. We also find that they have mesostable oils. But most of these have a relatively short half-life. The oil that resulted in the marshes were very stable, emulsified components, and that's the next level of refinement that I'm going to be talking about, is the very stable, emulsified oils. This is the reddish-brown material. It is stable from a few days to a year or better. It expands in volume instead of just having that oil sheen that's on the surface or that which is emulsified into droplets with surfactants. This actually imbibes water, forming capsules like in which the oil forms around water droplets. Viscosity increases dramatically in that it now becomes semi-solid. Before it was liquid as crude oil. And basically its density changes. It actually gets less dense or changes from floating on the surface to a density near the salt water. So movement of the oil now can be floating subsurface for long periods of time in a stable form that doesn't break down. And so it's a form that can be transported over long distances, which in this case is very important because we are 40 miles offshore for oil to arrive at the sites. The major component that allows this to happen is that the oil from the Deepwater Horizon site had a lot of asphaltenes. Asphaltenes are a group of planar type of aromatic compounds, large molecular weight, that when put into resin, they sort of coalesce at the water-oil interface and are deposited, forming an elastic material, a viscous material that sort of encapsulated the oil components, protecting it from further weathering, further degradation, and allowing it to be transported over long distances. So we have now a very stable form. a stable form that's protected from environmental degradation, a form that can be transported, and a form that, when reaching areas that are nursery areas and marsh areas, can coat surfaces. And so we have a emulsification that can present not just a gooey material, but potentially a transport modality of toxicity in a concentrated form, which is not affected by dilution of all that seawater that's found in the Gulf. So here we have a situation where the oil is coating grasses in areas that substrate breeding fish can interact associated with that. So we did a number of things, and this is just interaction to look at solubilization and show you a difference in solubilization between the emulsified and South Louisiana crude, which is essentially what the Mechanico or the Deepwater Horizon oil. On the left, we see a comparison with the South Louisiana crude with the concentration of the surfactant, the corexit, at the base. Let me just take this here. And we see increasing concentrations of corexit. The total amount of hydrocarbon, total crude oil dispersed, increased as the corexit concentration increased. The amounts that we see that were used vary because of surface application, but most of it is generally in this area right here. We see that the oil becomes solubilized with corexit. Now, weathering of oil, as what is known by managers of spills, prevents a lot of the solubilization or dilution of the oil product. And this is shown on this right graph. This is the emulsified, water and oil emulsified oil, such as what we've seen in the marshes, and we see that practically none of the oil is brought into the water, which one would expect, and this varied little with salinity in terms of the various types of product. So now we have a series of questions. We know that the surfactant isn't bringing that into solution. That material is being transported into areas where nursery functions for fish occur. What is the impact? Is this stuff even toxic at this point? That is the water and oil emulsion as it comes into play with these marsh grasses. So does the weathered and stable water emulsion present a hazard to developing fish? How does that relate to contact versus non-contact? Do the eggs from developing fish or a fish that in spawning have to come in contact with this material because it wasn't all over, or is it solubilized from coming into solution with this? What does the corexit alone, how does that influence development? Does it putting the corexit out? Even though most of the corexit was done offshore, what about even if it was at higher concentrations, would it have a developmental effect? And does corexit alter the emulsion effect upon embryonic development? So those are questions relating to what is the significance of this stuff other than impeding tarring and sort of things. Is it really toxic? We use the zebrafish as an animal model, developmental model, and there are a number of reasons representative of vertebrate development, rapid development, 6 days, practically all organogenesis after fertilization, 6 days post-have fertilization, that most of the development has already occurred. Very rapid. Large number, sexual dimorphism. The embryos are optically clear, so we could do various types of imaging in that. Very good model, but Are the zebrafish out in the environment? No. They're used as a model to examine the types of effects and significance of the exposure of the material that's reaching the marshes. So we were interested in this viscous sort of emulsified material. So we knew that it was surface coating the various types of vegetation. So we wanted to discoat material. So we decided on a dose relative to what the environmental loading was, and seeing that the direct contact exposure was by surface application. So they, in this case, the zebrafish eggs would be surface applied to the material, much like a surface or substrate breeding fish would lay eggs on the material. Then we also did the same sort of exposure, but suspended the eggs on a netting material above that, so it would be associated with waterborne exposure. So direct exposure versus waterborne exposure. This is just a graph of some of the effects. These are abnormalities in the embryos after hatch. This is concentration of corexit, increasing concentrations. And we have three groups. Corexit alone, that would be the surfactant or the dispersant. Non-contact or water exposure. Direct contact exposures, that would be the eggs right on the emulsion. And the very first thing I want to do is just pick out the corexit. Control would be no emulsion, no corexit, and then corexit concentrations as it increases. This would be the same as this for 0 concentration, 0.3, 3, and 100 milligrams per liter of corexit. You can see that the corexit alone, even at very high concentrations, concentrations that you would not see in the marsh environment and would only at the site of application see that it didn't cause developmental toxicity in the zebrafish at all. When we took and looked at non-contact exposure or water exposure, that is the egg suspended above that emulsion, that would be as if the eggs were around but not attached to the emulsion, we found that we did see an increase in the abnormalities in the developing embryo. But it was relatively small until the corexit concentration got higher. In other words, there was some component that was being elaborated from that emulsion that when in close environment would cause developmental abnormalities. But it only occurred when the surfactant was at high concentration, which in the situation of these marshes, 40 miles from where the spill and application dispersant was probably fairly unlikely to occur. Then direct contact, which we know happened in the marshes. Now we've seen that the percent abnormalities in the number of animals increased up to around 60% when the eggs were directly applied to these. Increasing concentrations of the corexit didn't seem to affect the total amount until at the very highest level of the surfactant, which again was not what would be observed associated with that. These are some of the developmental abnormalities. This is a control zebrafish embryo after hatch. This is cardiac edema, yolk sac edema. They have various lordosis and kyphosis and other types of spinal abnormalities associated with it. Just in comparison, this is from soot. The fluorescent shows these pHs are fluorescent. You can see in burned pHs what this looks like. This is from our direct exposure to the emulsion, and if the lights were off, you'd see just a very, very faint blue. So a lot of the exposure was very low, and non-contact exposure, nothing even observable with the lights off. So how did this affect mortality? Same sort of exposure or thing, but just following those animals out longer, the abnormalities, and What we've seen, again, the surfactant. For exit, nothing. The waterborne exposure, really these were malformed, but during our period of time that we were looking at, 192 hours, not significant number of mortalities, but direct contact exposure, you can see the mortality rates were very high associated with this. So, thinking about another question about exposure, does the developmental toxicity of direct contact with this material influence the animals on a dose-response basis? In other words, if we had a very thin layer, which we had, 250 milligrams over 16 square centimeters in this, which simulated dosages, if we increased the thickness, did it affect it? And the question really came as a function is in veterinary medicine, when we apply radiation therapy to tumors, we know it's by a surface area related phenomenon rather than a dose phenomenon. It's a surface area thing. So we examined that. And what we found is that even by increasing the dose dramatically, we find that both the abnormalities and the number of dead were not significantly different over a fourfold dosage increase. So it's thicker emulsion on that plant material really wasn't that significant until we started to see some increased mortalities when we had an eightfold increase. So for the large part, not stoichiometric. So no matter how thick that oil was on that plant material and how the eggs of these fish were being laid and had very small pinpoint interactions with that. And the thickness of the material didn't make it that much more toxic. It was even very thin layers was almost as toxic as very thick layers of the material. Another question, will chronic air exposure affect the toxicity associated with it? Well, tides go in and out. As the tide goes out, the material is subject to the heat of the sun, the drying action. Does that affect the toxicity? And so what was done is that, again, cups were buttered, put in material like that, and basically allowed to dry for a period of time, and then toxicity examined. Again, very simple. Again, most of these things we had gene-related profiles that we also followed. And we found that the oxidized material versus freshly exposed material was nearly as toxic over a 9-day air-dried procedure. So this is time before it undergoes degradation, but it was still a time where you expect that some physical processing events would influence this. Another question. If there's a critical exposure interval, do these embryos during development have a particular period of time where these developmental abnormalities occur in terms of that exposure regime? We looked at 0 to 48, I'm using emulsion again, 48 to 96, 96 to 192. And the period where the most observable effect, again, this was zebrafish and zebrafish development. Fish vary in their developmental times in terms of dealing with this, was 0 to 48. And that occurs right before hatch. They would hatch at about 48 hours. So once they became free swimming, and exposure was at free swimming, less of an issue. Maybe an exposure issue, maybe a sensitivity issue. Does the Deepwater Horizon emulsion that reaches have prolonged toxicity? So what we did was we took cups, applied it onto the surface of these cups, put the cups under flowing water for extended periods of time, seen observable degradation of the emulsion, gently rinsed off any particulates off it, put embryo media on, put freshly fertilized eggs on it after the period of washout. And the periods of washout were 0 43, 167, 236, and 328 days of washout, continuous flow through water. So given that that oil spill occurred and the material got into the marsh and is on this material, how long was that still toxic to the embryo? And this shows a variation with that, but this is percent abnormalities. These are their controls. We see some variability, but low levels of developmental abnormalities in control. And this would be equivalent to the first day exposure of fresh material. This is 43, 167, 236, 328 days after washing this material with water, then putting freshly fertilized eggs after that washout. Could that still elicit developmental abnormalities? And even at 328, where about 36% of these animals were showing developmental abnormalities. And if you physically looked at the emulsion, it was degrading. You could see worm pilings on it from material. You could see it looked sort of that brownish color. It was sort of an off-green gray. It was spotty in some parts, but still had developmental toxicity, which suggested that this material has long-term implications in terms of developmental toxicity. Now, does that mean that the fish out there? No, they're not zebrafish, but we're looking at the emulsion capability of the emulsion and processing event with that. One of the aspects of this was hatching delays with the embryos. We've seen a significant delay in hatch. So why was this occurring? So this is an embryo in the chorion, in the egg, before hatch. This would be equivalent to pre-48 hours. And so if we looked at hatch with that percent of hatch relative to control or the emulsion-exposed animals, at 48, you see it's about 60% of the controls were hatched and less than 1%. Out to 72 hours, 100% of the controls, or nearly 100%. We had, I think, 3 out of 16 mortalities out of that, with less than 5%. So it's definitely delayed in terms of the hatch environment. Doing SEM and TEM on these, we looked at the control, and just for orientation, this would be the embryo. This would be the egg casing. This is at 48 hours right before hatch. So these embryos haven't hatched yet. The chorion is still around it, but time-wise, they're just about ready to be hatched. This is the para-embryonic space, the embryo, the egg caps the chorion, and you can see how the chorion and scalp, the chorionases, which are produced, cause dissolution of the chorion so that the embryo can kick and break the chorion and get out. If we look at emulsion, again, the delay, we looked at 72 hours. Here's the embryo. This is the para-embryonic space equivalent to this, filled with an electron-dense material. And if you look at the whole gist of the chorion, it has much less chorionase activity, suggesting that either the enzyme is inhibited or can't be brought to the bear in terms of that hatching protocol. These are the hatching glands and some of the chorionases that are involved with coming to a surface. And again, coming to the surface, it appears that the material was being brought there, but wasn't either active to allow for the hatch. Now, many of the embryos, even with the water and oil emulsion, showed they eventually hatched, but would have developmental abnormalities. But the whole process was slowed dramatically by what we believe is some of these processing events. So this is sort of just a brief overview of some of the exposure data. And the purpose of giving this information is just to show that some interesting questions can be addressed by very simple means sometimes. We tend to get involved with a lot of our sophisticated interactions, but some very elegant studies. You go back through the literature and you look at some of the elegant work that's been done, it's the question that's very important in terms of dealing with some of the answers in terms of this. So what conclusions can I draw from this information? The Deepwater Horizons definitely are developmentally toxic to the zebrafish embryo. The dispersant doesn't seem to have much of an effect until you get a very high concentrations, which were never attained in the environment. Direct contact exposure versus waterborne exposure is definitely, for substrate breeding fish and eggs, is definitely much more toxic. The developmental toxic is non-stoichiometric with direct contact. Again, that surface area phenomenon. And we don't even know if it's the elaboration of toxic materials from that and transfer, or a physical process, such as membrane changes that may be involved with that. Air-exposed water emulsions retains developmental toxicity. There is an interval where post-fertilization, which appears to be most sensitive, though this may relate to the actual hatching process and involvement with that. Water and oil emulsions can act as a transport modality. I remember watching news one night on, was it CNN or ABC? I don't remember. And there was a guy walking along the beach and pulled out these clumps of moose on the beach in Pensacola and said, other than an eyesore, these things aren't hazardous. Well, those materials are water emulsions that basically, if you broke them apart and surface applied on material, it could elicit this developmental toxicity. So packaging and shipping, it's basically shipping toxic materials over a long distance in a form that's very resilient to degradation. Prolonged washout, even when thinly applied, that stuff is long-term toxicity, and that we've seen that issues and variability changed, and whether that's experimental or other related interactions, we're not sure. So I was given the task to give a little bit of a career path things, and I was in a similar situation many years ago, and I had a summer fellowship through National Science Foundation. So I always like to sort of speak to that issue. I was interested in animals and outdoors, but there really wasn't that many opportunities. So I decided to pursue veterinary medicine because of the animal component of it. And so I was an undergrad in zoology, okay, interactions with that. I took an elective in invertebrate zoology, met what turned out to be my major professor during my PhD, and I really liked invertebrate zoology. I liked the aquatic related issues that were largely emphasized because he was a limnologist, lake studies person. And so I thought that was great. And he said, well, during class that they had an NSF summer fellowship research program. And if I was interested, I should apply. Well, I applied and had that, a similar experience as you're having this summer. And it was regarding chlorine toxicity with power plants to fish. They use chlorine to biodefoul their cooling tubules. And I really got into it. I was very interested in the type of things. So now I had veterinary medicine as an interest, environment, aquatic tox, research. I was totally confused. I still am totally confused. And so I still applied to vet school because I had put everything. I didn't get in. Big surprise. When I found out later, when I did finally get in, they had we had 2 people out of a class of 80 that had only applied once. Everybody had applied twice or 3 times to get in. This was at the University of Minnesota. So I didn't get in. I pursued a master's degree in aquatic tox, reapplied, didn't get in again. Oh, I was feeling pretty down in the drums about this, but I really felt really good about this other because it was very interesting to me. Then I entered a PhD program in aquatic biology, and we had an interesting program there. They had an NIEHS Aquatic Biomedical Center and the Medical College of Wisconsin and the Center for Great Lakes Studies, which all I developed into a hybrid program associated with the aquatic toxin environment. Reapplied to vet school. I remember filling out the vet school application by hand about an hour before it had to be in the mail. Sent it in. I was admitted to vet school, go figure. With the other ones I had typed, and it was all nice and everything. But along the way, as I worked through that, I had realized that I was becoming more and more competitive with the more degrees. There was 11 people in my veterinary class that had masters or PhDs. So it was definitely something that they were looking for. And then added more coursework in aquatic animal medicine. I became interested in that. Completed vet school. Went back to finish my PhD. Completed the PhD. Then I did some advanced training at Woods Hole Oceanographic through Cornell and Penn for aquatic animal medicine, and did a 3-year postdoc in aquatic toxicology. So what I'm saying is, if I thought about doing this up front, I wouldn't have done it. It just sort of happened because of the interest. But it all stems, this sort of confusion, of what you're doing this summer. And that research experience may open different avenues. And I would suggest to you that you take as many different opportunities that are available to you to explore what your inner interest is. And if anybody has any questions, I'd be glad to do that. Yeah. The chorion? It's harder to hatch because the chorion isn't being dissolved. And so the embryos normally, what the chorionase, the enzyme that is secreted by the embryos, it puts these scalpings in that. And so its spacings are thinner, so when it kicks, it breaks. Well, with the ones that had the water and oil emulsion, that barrier was almost intact. So they'd kick against it and couldn't break it. What? I'm sorry, I missed the first part. We didn't follow that, but other studies have, and there has been some evidence showing some second generational effects, but most of these animals that showed abnormalities died. We were only looking through the first 192 hours of development. And so the other thing that you have to keep in mind with this is that these animals that have abnormalities, if we're thinking about an animal out in the real world, what would that mean? That means you're food for somebody else. You know, because they're not able to have the fitness to survive. Then you're all SOL. Yeah. I'm sorry, I have a little yeah, they wouldn't be able to swim or do anything, feed very well or anything. Is that answering your question? It depends if they were, the embryos that have a very limited, would have a very limited range. Again, zebrafish are model, but if we had fish that were in the area, these small fish would have very limited range. A larger predator may range in and out of exposure areas. So they may be totally fit. They may be moving in or out of oil exposed areas. Oh, would they be poisoned by that? Yeah, potentially, yeah. In fact, most of the work that I've been associated with is food chain transfer of contaminants. And a lot of these pHs are not only metabolized, but they're metabolized to more toxic intermediates, which are then ingested by subsequent parts of the food chain. Well, they're not readily biomagnified, but they're readily activated. Yeah. Well, definitely the way that the animals, again, I dealt with substrate breeders, but breeders that would put the spawn out in a pelagic environment, if it didn't have the direct oil exposure because of dilutional phenomenon, probably much less effective.
Okay. Any other questions? Folks from the remote sites? I can see it. Is that a hand up or are you just combing your hair? Sorry, it's hard to tell.
Or the person sleeping there back in the corner. No.
Just a joke.
That's right.
Please help me thank Dr. Kleinow again.
Thanks.